Hello everyone and welcome to LabX webinar. I'm Laura Quevedo and I'd like to welcome you to today's webinar, Unlock the Full Potential of RNA Sequencing with Twist Bioscience. We encourage you to submit your questions to us at any point during today's webinar. To ask a question or make a comment, simply type your question into the Q&A box, which is located on the right-hand side of your screen. I will forward any unanswered questions to today's speakers and they can respond to you directly if possible. Additional resources for today's presentation are also located in the Handouts section on the right-hand side of your screen. I would like to remind you that this webinar recording will be available on demand shortly following this presentation. So please watch your emails for information from LabX on how to access this free video. I would especially like to thank Twist Bioscience for sponsoring this event. This support allows LabX to keep these webinars free of charge for our readers. With that, I would like to introduce our presenters for today's webinar. James Flynn, Product Manager at Twist Bioscience, and Damon Anderson, Technology Editor for LabX. Thank you both for joining us today.
Welcome. My name is Damon Anderson, and I'm the Technology Editor at LabX. We're happy to showcase the first in a series of Q&A sessions with Twist Bioscience. In today's session, we will be highlighting the Twist RNA sequencing workflow solution with expert insight provided by our guest, James Flynn, PhD. Welcome, James.
Thank you for having me.
Pleasure to have you. James is a seasoned biotechnology product leader with a strong track record in advancing genomic technologies for both clinical and research applications. At Twist Bioscience, he serves as a staff product manager overseeing next-generation sequencing target enrichment solutions that support precision oncology and translational research initiatives. And these include partnerships with long-read sequencing applications. So RNA is fundamental to how our cells function, translating our DNA blueprints into functional molecules, which ultimately help build and regulate the proteins which make up our tissues. RNA sequencing is fundamental to the understanding of dynamic biological processes. So James, can you tell us more about the critical role of RNA analysis in biological investigations and the importance of RNA sequencing in disease research?
Yes, I'd be happy to. RNA is really an interesting area compared to DNA. DNA, you can imagine your genome is fairly static. You have that sequence in every cell in your body, whereas RNA is very dynamic and it can change at any time under different conditions or in response to different stimuli. So RNA sequencing enables a number of areas that are very important, including functional studies, where a genome may be constant, but then it's under different experimental conditions, which can have a profound effect on gene expression. And the expression of genes is ultimately regulates our cells, and it also provides the template for producing the proteins that make up all of our tissues. So one example would be to have a wild type mouse and then knock out a particular gene and then look at the different effects that might have. And the mode that we do that through is through RNA sequencing. So another area that is very useful for RNA sequencing is to predict the transcript sequence from the genome sequence itself can be very difficult. Each gene has different isoforms and they can be rearranged to produce different RNA molecules, which leads to different proteins that are made from that. It can also impact how that gene actually regulates the cell. And then the third area is that some molecular features can only be observed at the RNA level. As I just mentioned, alternative isoforms or perhaps two genes become fused together, and that may drive some disease like cancer. And then there's also the ability to edit RNA, where we can actually artificially change the sequence of RNA. So all of those studies are enabled and are much more useful with the ability to do RNA sequencing to look at all of the genes being affected all at the same time.
Great, very informative, and thank you. can you describe the capabilities of the Twist RNA workflow solution for a range of possible applications?
Yes, as I just presented in the previous slide, there's a number of different use cases for RNA sequencing. But some of the areas that Twist is most focused on is looking at areas such as oncology, understanding genetic disease research, how a gene may be mutated in a cell, and you want to understand exactly why that causes the disease. Twist also makes solutions for infectious disease research. As you know, some viruses are based in DNA genomes and some have RNA genomes, and so you'd want to do RNA sequencing on those. There's other applications such as cell and molecular biology research, fundamentally understanding how cells work. And then things such as RNA therapy research, which kind of is working on future therapeutics that all is empowered through RNA sequencing.
Can you provide details of the typical RNA processing procedure with regard to background depletion, fragmentation, first strand synthesis, double strand synthesis, and so forth?
Yes, I'd be happy to. What I'm showing you here is just an overview of how RNA sequencing works using our workflow. So it all begins with sample preparation, where you'd start with extracted RNA material. That can go into our RNA library prep kit. And there's an option there to also do ribosomal RNA depletion and hemoglobin depletion. Those are basically expressed genes that most people aren't very interested in, and it actually costs sequencing reads. So we always offer the option up front to deplete those or remove those from the library before going into library prep. We also offer the option to include unique molecular indices, or UMIs. It's essentially just a barcode for each molecule, which tells you where did this sequencing read come from, and can you collapse that back down into just the reads, and you want to understand more accurately the expression of genes. And then what Twist does, and really what we excel at, is we have the solutions for target enrichment for RNA. So once you make your library prep and you complete that first step, you have what's known as a whole transcriptome library. It captures all of the RNA molecules one might be interested in. But sometimes that's a bit too much information, and it actually costs more per sample to do. So what we can do is target enrichment, and makes it more efficient to just focus in on the things that you're interested in that RNA library. So we offer a couple of solutions there. We have the RNA exome, which is a whole transcriptome enrichment panel. We have the RNA fusion panel, which is just focused in on oncology-related gene fusions. And then, of course, Twist can make any sort of custom panel. So we make any sort of RNA panel that you might be interested in is also available from Twist. So this is just showing you another way to look at that. So I showed you on the left side here, this is our whole transcriptome workflow. Again, you can have the option to do the depletion module up front of that if you're working in some samples such as blood-based samples that might have a high degree of hemoglobin being expressed in them, and ribosomal RNAs, which are just ubiquitous in samples. So that can be depleted. And then we have our whole transcriptome library prep kit. And that is exactly the same library prep kit that you'd start with if you're doing the target enrichment workflow. The target enrichment workflow just adds a couple extra steps to enrich just the portion of the library that you want, and that is paired up with our target enrichment panels, or TE panels, as we like to say. Next, you also asked about how the workflow proceeds and how do we end up with a sequencing-ready library. So as you can see, this is a little bit hard to see size-wise, but I can walk you through the steps on the right here. You start with a total RNA sample, and again, you have the option to deplete molecules out of that sample up front of the library prep. We then use heat and digestion with enzymes to fragment all of that RNA into shorter fragments that are sequenceable, because most sequencing platforms and short read sequencing can't do beyond about 100 bases, 150 bases. And so what we do is that fragment that material. From there, Twist's kit does a first strand synthesis where it reverse transcribes the RNA molecule into a cDNA molecule. And then from there, in the second strand synthesis, it completes or makes the complement to that DNA strand. And then in the next step, we do an N repair where a phosphate's added, and then an A tailing where an A base is added at the end. And then that prepares the library for adapter ligation, where we can use Twist either UMI or universal adapters can be ligated onto those molecules. And then from there, now you have a complete sequencing ready library. I would just note that Twist workflow is really focused in on having a very simple workflow and one that's fast. Those were basically the two criteria that Twist was going for. So we have a very fast single day workflow where Twist has combined certain steps in the workflow into one step, meaning less hands-on time for you as you're preparing your libraries. We've also eliminated some bead purification steps in the workflow to make things, again, less hands-on time, faster to get your sequencing ready library. And finally, we wanted to make this automation friendly. So should you have high throughput, we can definitely add automation on top of this as well.
So perhaps you can elaborate on the performance of the Twist RNA library kit for whole transcription analysis.
Yes, I'd be happy to. I just explained that we were really focused in on speed and the ease of use of the kit, but it's also very high performing as well. So what I'm showing you here is a series of comparisons using either universal human reference RNA, which is just basically a standard material that we use, or an FFPE, which is formalin fixed paraffin embedded material. And I'm showing you from as little as 1 nanogram. In the middle, it's 10 nanograms or 100 nanograms. You can see the concordance that you can get from run to run. It's highly reproducible. And then on the left side, I'm just showing you based on the amount of input from as little as 1 nanogram up to 1,000 nanograms, which is the range of input for the kit. You can see how many genes are detected across those for both reference RNA, which is very high quality, or FFPE, which is typically more degraded and a more difficult sample type. We still have very high performing results.
Yeah, so it's amazing that the FFPE performs as well or similar to the reference data sets. Great. Excellent information. Can you describe the use of the Twist RNA sequencing probe for RNA exome sequencing and elaborate perhaps on the performance of the kit for exome enrichment?
Yeah, I'd be happy to. What Twist is designed, as I mentioned, there's our specialization is really in target enrichment, and what we've designed is something kind of unique to the market. Is we've used something that we call exon-aware design. As you can imagine, there's different exons within a gene that are expressed into an RNA molecule. And we designed our probes intentionally, placing those probes so they never cross an exon-exon junction. And what that can happen is if you have a probe that crosses that exon-exon junction, you could potentially pick up off-target molecules that you're not really interested in. The other thing with the exon-aware design is that if you place the probe just next to the exon-exon junction, is that you can potentially pick up novel rearrangements of the gene. And that includes things in oncology where you might have a gene fusion transcript, and that's very important to understand if there's a gene fusion event. So we've built this panel to cover all the genes in the genome and every isoform of that gene. And so that allows you to do differential expression and variant calling and gene fusion all from the same sample. And at the same time, you can also do this with actually less sequencing than you do in a whole transcriptome sample. So it's more efficient, but it also allows you to do some additional things like variant calling. This also is especially important in FFPE samples. We actually get better enrichment. So if you start with a worse sample, RNA exome actually provides better enrichment of those samples as compared to whole transcriptome sequencing. So yeah, again, as I mentioned, saving sequencing costs, and it works better in FFPE, and it allows you to do variant calling off of RNA. So this is just another slide showing you what I just mentioned about the enrichment is that if you start with really high quality RNA, you get about 1.8 to 1.4 fold enrichment versus an FFPE sample, you get greater than 2.4 fold enrichment on average is what we see. So the plot I'm showing you in the bottom left is if you took a whole transcriptome sample with 10 million reads, and ask the question, well, how many number of genes can I detect with that 10 million reads? It's a little bit under 15,000 genes. And then you compare that to doing the RNA exome, you can see with just 6 million reads, we get the same amount of information out of the sample. So it's saving you a substantial amount of sequencing costs, and you're actually getting the same amount of information. And on the right, I'm just showing you a comparison of Twist RNA exome with the exon-aware design as compared to a competitor's RNA exome. We get about 20-ish% more on-target reads, and that's going to translate to maybe things that you might have missed with another capture panel.
Excellent. Thank you very much. So you've given us some great comparison data. Perhaps you can compare the Twist RNA exome sequencing to poly-A sequencing in terms of performance and efficiency.
Sure, that would be happy to make that comparison. So poly-A sequencing for RNA is a means to capture RNA molecules that are already polyadenylated, meaning it has a series of A bases at the end. And that tail end that has the series of A bases is what's used to capture an RNA molecule that's been fully spliced and polyadenylated. Whereas RNA exome, we're placing the probes and we're capturing all the way across that whole transcript. So if you look at the bottom plot on the right, you can see that with poly-A sequencing, you're capturing it based on the tail end of the sequence. So most of the sequencing that you get is positioned at the end of that transcript. And sometimes that's good enough to just identify the transcript, but maybe you want to know more information about, let's say, the particular exons that are in that, or you just want the full sequence of the RNA. You don't get that with poly-A sequencing because there's a bias towards the end of the transcript. But with Twist RNA exome, you get much more even coverage across that. And then what I'm showing you above in that bar plot is that you get far more of the sequencing reads are in the coding sequence of the gene. And so if you're interested in that, you wanted to call variants, let's say in particular, you could do that with RNA exome. So the bottom line is that it's just a more efficient capture and provides more even coverage across the whole RNA molecule.
Yeah, even coverage, less bias. I completely get it, and it's just amazing. It's amazing data that you're showing us. Great. So, have there been any collaborations in which Twist RNA workflow solution has been used or implemented as a diagnostic or screening program? Perhaps you can describe one such program that you've worked with.
Sure. Twist, we work with a number of customers. They're doing a bunch of different applications. So I'm actually the product manager that's in charge of our Alliance Panel program. What that means is we actually partner with some of our customers who have developed unique content. One such customer was CGAT in Europe, who's a diagnostic company. They have licensed their content of their panel. So this is CGAT's design for cancer-related fusions. So there's 160 known fusions in the panel, and that covers 30 different indications of cancer, and also includes more 80 of those fusions are therapeutically actionable, meaning the results that you would get from running this panel, you could potentially do this to identify potential therapeutic interventions. I will mention that this panel is for research use only. And so this is basically a tool for research. It is not for diagnostic purposes. But that being said, fusions are incredibly important because it indicates that there's been some sort of chromosomal rearrangement. Two genes that shouldn't be together are placed together and they make a RNA molecule called a fusion transcript. And that's what actually could be a driver of the cancer or the tumor. So really important panel, and we're happy to partner with some of these groups to offer this content more broadly.
Can you describe the key metrics to consider when assessing RNA sequencing workflows, specifically hybrid capture versus fusion detection?
Yeah, there's different methods for investigating RNA out there. We obviously feel that our solution is really one that offers the customer the greatest flexibility and performance. So I just wanted to have this quick comparison of what hybrid capture can do or target enrichment can do versus amplicon-based sequencing. So with hybrid capture, it's very easy to customize the content. So if you wanted to add additional fusions or you had certain genes you wanted to cover or not cover, that's very easy for Twist to do. We customize panels all the time. Whereas amplicon sequencing, you're actually designing primers to those target regions. And it's important that all those primers work together properly and they don't pick up, let's say, an off-target amplicon where two primers that aren't supposed to work together actually produce something, and that can lead to spurious results. Performance-wise is that Twist makes double-stranded DNA probes to capture these libraries. It results in really, really uniform coverage of those targets. And then Twist, on top of that, has a number of design strategies we can do to boost the coverage of genes or perhaps tone down the coverage of certain genes. Whereas I mentioned with amplicon sequencing, you can lead to things like maybe an allele drops out, meaning like it didn't amplify properly. Or as I mentioned, you could misprime and pick up something that you don't want to do. Finally, there's the discovery aspect of this is that with our target enrichment, if you recall to RNA exome, we've designed those probes so it can pick up novel things. So you can pick up fusion events, let's say, that are known events, and you're looking for those specifically. But you can also pick up novel fusions at the same time because the probes are placed right on that exon-exon junction. So if there's something that is new that has never been discovered, you could pick that up. Whereas amplicon-based sequencing, you need to know the target in advance for the most part. And I'll give the example of that anchor multiplex PCR. To get to that novel discovery, you have to use way more input. So on the order of 200 nanograms is recommended input, whereas our kit takes as little as 1 nanogram and can do the same thing.
This is great. We've covered a lot of ground. So at this point, perhaps you can offer us a recap of the key features and benefits of the Twist RNA sequencing solution as compared to other conventional solutions that are out there in the market.
Yeah, I just wanted to remind everybody that we do offer both whole transcriptome sequencing and target enrichment sequencing, which can lower your sequencing costs. It can enable you to do things like variant calling. We also can customize any of those solutions however you want. But I just want to remind everybody that our library prep, we're really focused on making it the least amount of hands-on time and having a single day workflow. We have flexible and modular kits, so you can pick up the components of the workflow that you need. You don't have to buy an all-in-one solution. So however your research is going and whatever samples you're using, we have solutions for you. As I mentioned, it takes a wide range of input and quality. I showed you some data on FFPE, as well as that our kit can input as little as 1 nanogram of material all the way up to 1,000. And all of this is automatable at whatever device that you might have. We have a team of automation FAS that can help you set up your lab to do this in a high throughput manner. And finally, we're quite innovative in how we're designing these RNA panels with our exon-aware designs.
So with that, we'd like to thank our audience, and thank you, James, for joining us to speak about the Twist RNA sequencing solution.
Yeah, happy to, and thank you for your attention.
For more information, please visit the website displayed at the bottom of your screen. And we'll be back soon with the next installment of the Twist application series featuring a Q&A session focused on the Twist cfDNA sequencing solution. Thanks very much. Have a great day.
Thanks, Damon.
This concludes our webinar. On behalf of LabX, I'd like to thank James Flynn, Product Manager at Twist Bioscience, for his insight, as well as LabX Technology Editor Damon Anderson for facilitating this conversation. I would like to thank Twist Bioscience, whose support allows LabX to offer these webinars free of charge for our audience. For more information on all our upcoming or on-demand webinars, or to learn more about the sale and purchasing process for scientific, process, electronic testing, and medical equipment, please visit our website at labx.com. We hope you can join us again soon. Thank you and have a great day.